Hello everyone, myself Shabir Ahmad Magray, student of M.Sc in the division of Food Science. Today I am going to deliver my presentation on understanding the principles of biodiversity and strategies in germplasm conservation of coffee. Before starting, I would like to thank Dr. Prashant Bakshi sir for giving me this golden opportunity to share my views regarding this topic. Here is my topic: understanding the principles of biodiversity and strategies in germplasm conservation of coffee. Here are the contents related to my topic. First, biodiversity. Second, general view about coffee. Third, center of origin of coffee. Fourth, morphology. Fifth, adaptation. Sixth, biochemical components. Seventh, biodiversity conservation. Eighth, insight into coffee genetic diversity. Ninth, conservation options. And tenth, in situ conservation. 11. On-farm conservation. 12. Conservation in field gene banks. 13. In vitro conservation. 14. Pollen storage. 15. DNA storage. 16. Seed storage. 17. Cryopreservation. 18. Cultivars. 19th. Conservation of coffee germplasm. 20th. Geographical indication certification for 5 varieties of Indian coffee. 21. Intellectual property rights. Biodiversity. Biodiversity is actually the combination of two words, that is, bio means life and diversity means variety. Thus, it is the variety of life on Earth. Term biodiversity was coined by Walter G. Rostand. Levels of biodiversity. Biodiversity is usually explored at three levels: genetic diversity, which means variety of genes in a species; species diversity, which means variety of species within a habitat or region; ecosystem diversity, which means variety of ecosystems in a given place. General view about coffee. Coffee, coffea species, is one of the most important beverages in the world and is consumed by more than a third of the world's population. Very important commodity crop for many developing countries, once contributing over US 10 to 11 billion dollars annually, providing a source of income for thousands of small-scale farmers, as well as being a significant source of employment. The commercial coffee comes from two main species, Coffea arabica and Coffea sinaphora. Pests and disease such as coffee berry borer, coffee berry disease, coffee leaf rust, and more recently, Fusarium wilt and others undermine coffee production and quality. It is recognized that the cultivated varieties, in particular Coffea arabica, have a very narrow genetic base, and their improvement depends on availability of adequate amounts of genetic diversity. Genus Coffea is endemic to the Old World tropics of Africa, particularly Madagascar, and over 100 wild species are found in Afro-tropical Madagascar region, including the Comoros and the Mascarene Islands. However, deforestation and encroachment by agricultural activities, population pressures, and economic hardships threaten all these reservoirs of genetic diversity. And with these threats comes the danger of significant erosion of the coffea gene pool. The conservation of coffee genetic resource has not received much attention recently, but efforts to collect and conserve coffee genetic resources were initiated in the 1960s and 1970s by Orston, now we called it IRD, FAO, and IBPGR, now Biodiversity. Previously, it was called the International Board of Plant Genetic Resources. Now we call it International Biodiversity of Plant Genetic Resources, and several options for their conservation have been developed. Centers of origin. A center of origin or a center of diversity is a geographical area where a group of organisms, either domesticated or wild, first developed its distinctive properties. They are also considered as centers of diversity. Center of origin were first identified in 1924 by Nikolai Vavilov. Coffee grown worldwide can trace its heritage back to centuries. The ancient coffee forests on the Ethiopian plateau. Morphology. There can be a wide diversity among morphological traits in coffee plants. Coffee plants can be shrubs or trees whose height varies from 1 meter in Coffea humilis up to 20 meter in Coffea liberica var Daviri. Leaf size varies considerably from 2 centimeter, for example in Coffea eugeenoides, to 48 centimeter. Example is Coffea magnificula in length and from 0.8 centimeter to 30 centimeter in width. Mature fruits are generally red. Example Coffea arabica and Coffea sinaphora, but also yellow. Example Coffea liberica. They can also be orange, for example Coffea congens, or violet Coffea racemosa, or black, example Coffea salvatrix. Further, white fruits were observed in Silenthes taxon collected in Congo. Adaptation. Altitude is an important factor in structuring forest habitats in Africa. The coffee species are distributed from sea level, example Coffea liberica or liberica, to 2100 meters, example Coffea eugeenoides. Among the cultivated species, Coffea sinaphora grows in lowland forests up to 1400 meters, while Coffea arabica is found in sub-montane forests between 1000 and 2000 meters. These ecological differences of habitat could explain the variations observed in cold sensitivity among in vitro microclimates of different species stored at various temperatures. Biochemical components of the numerous compounds found in green coffee beans. Attention was firstly focused upon caffeine because of its known pharmacological actions and influence on beverage bitterness. Caffeine content of cultivated species appears moderate in Coffea arabica, varying between 0.76 and 1.82% dry mass basis percent DMB, and high in Coffea sinaphora between 1.51 and 3.33% DMB. Caffeine-free species were reported in Madagascar region, Eastern Africa, and Central Africa. Other aroma precursors have been studied, such as chlorogenic acids, which increase bitterness of beverage, and sucrose and tritronellene, which give rise to appreciated flavor products. Biodiversity conservation, the protection and management of biodiversity to obtain resources for sustainable development. It has three main objectives: sustainable utilization of species and ecosystem to maintain life's supporting system, and third one, essential ecological processes. Biodiversity International is an independent international scientific organization that seeks to improve the well-being of present and future generations of people by enhancing conservation and the deployment of agricultural biodiversity on farms and forests. Financial support for biodiversity research is provided by more than 150 donors, including governments, private foundations, and international organizations. Insight into coffee genetic diversity. The genus Coffea is characterized by a large number of species whose differentiation has occurred relatively recently. It is thought that the diversity of coffee species and the genetic diversity within them have been the result of rapid speciation and adaptive radiation process. Diversity can be analyzed at the genetic and morphological levels, the latter often being used as a proxy of the former. Only few studies have been undertaken to examine the levels of genetic diversity in wild coffee plants. Bean size is another variable character which makes necessary the definition of an adequate desiccation time prior to cryopreservation. Extreme values of 1000 bean weight were 29 grams, example in Coffea pseudogingivari, and 198 grams, example Coffea liberica. Conservation options. Two basic conservation strategies, each comprise various techniques, are employed to conserve genetic diversity, namely in situ and ex situ conservation. Article 2 of the Convention on Biological Diversity provides the following definitions for these categories. Ex situ conservation means the conservation of components of biological diversity outside their natural habitat, while as in situ conservation means the conservation of ecosystems and natural habitats and the maintenance and recovery of viable populations of species and in the case of domesticated or cultivated species in the surroundings where they have developed their distinctive properties. In situ conservation. In situ conservation offers the possibility of conserving a greater diversity of species and gene pools at the same time. It's a dynamic conservation process as plants continue to evolve with changes in their environment. Most importantly, pests and diseases. It is suitable for crop evolution and genetic studies and represents a viable alternative for conservation of non-orthodox seed species. However, in situ conservation leaves the plant material vulnerable to natural and human-induced disasters, and the plant material is not rapidly accessible for use. The appropriate management regimes are poorly understood, and high levels of supervision and monitoring are required to implement in situ conservation. On-farm conservation. On-farm conservation is also a dynamic process in which plants continue to evolve. It ensures the conservation of valuable genetic diversity in traditional landrace, weedy crops, and ancestral forms. Conservation in field gene banks. Conservation in field gene banks is suitable for species with non-orthodox storage behavior. In some ways, this method offers a satisfactory approach to conservation. The genetic resources under conservation can be readily assessed and observed, thus permitting detailed evaluation. In vitro conservation. In vitro conservation methods represent a relatively easy alternative for medium to long-term conservation of large number of non-orthodox, sterile or colonial species. Germplasm exchange is facilitated by in vitro methods as they permit the production of virus-free material through meristem culture and their rapid multiplication. However, there are risks of somoclonal variation in some species when maintained under in vitro slow growth. In vitro storage is relatively high-tech and maintenance costs of the material are high. Pollen storage. Pollen conservation is another viable alternative for conserving species with non-orthodox seeds. Pollen storage is relatively easy and low-cost procedure. Using pollen facilitates germplasm exchange as a relatively small quantity of material is required for a single sample. More importantly, pollen is generally less likely to be infected by pathogens than other propagules. The disadvantage of pollen conservation are that only paternal material is conserved, that is less than half of the total genetic makeup of an organism. DNA storage. DNA storage in DNA library is yet another alternative for conserving species with non-orthodox seeds. It is also a relatively easy and low-cost procedure. It is particularly useful for conserving specific genes responsible for heritable characteristics of particular value, example, disease resistance. DNA is easily accessible and is especially convenient for exchange among plant breeders. However, procedures for regenerating entire plants from conserved DNA are not available at present, and numerous problems exist with gene isolation, cloning, and transfer. Cryopreservation, the traditional ex situ conservation method for these difficult to store categories of plant is in the form of field collections, which present both advantages and major drawbacks as described previously. Cryopreservation, that is the storage of biological material at ultra-low temperatures, usually that of liquid nitrogen at -196 degrees Celsius, is the only technique currently available to ensure the safety and cost-efficient long-term conservation of genetic resources of the problem species mentioned above. Conservation of coffee germplasm. The conservation of coffee germplasm is closely associated with coffee arabica domestication and has predominantly involved conservation in field gene banks because of the non-orthodox nature of the coffee seeds. It first began on farms in the center of origin, that is Ethiopia. Subsequently, worldwide extension of coffee cultivation contributed to the establishment of field gene banks in producer countries. The size of the collections increased greatly during the second half of the last century, when coffee germplasm collections were made during explorations across Africa. Considering the lifespan of coffee trees, about 30 years, and the inherent problems associated with maintenance of field gene banks, as described above, there is an there is now an urgent need for regenerating the aging coffee trees and for the development of complementary methods of conservation. Numerous in vitro techniques have thus been developed for medium-term storage of coffee germplasm. The establishment of an in vitro coffee core collection was initiated in 1991 at IRD Montefiore, France, but a few years later the limits of this technique was recognized with the occurrence of some genotypic selection and intraspecific genetic drift. Major coffee gene banks in the world and distribution of the coffee arabica accession collected in Ethiopia by FAO in 1968 and Orston surveys area. It was actually Latin America. The three countries, that is Costa Rica, Colombia, and Brazil, institutes which were involved. It was CATI in Costa Rica, CENICAF in Colombia, and IAC in Brazil. FOA collection in Costa Rica was original, while Orstown doesn't collect anything from Costa Rica. Colombia FOA collection was duplicate, while its Orstom collection was also duplicate. Duplicate collection simply means the wild relative or the relative of the original one. Brazil. In Brazil, FOA collection was original, while its Orstom collection was duplicate. In Africa, the FOA collection was duplicate, and Orstom collection was original. In Asia, the country was India, while the FOA collection was original, and Orstom doesn't collect. Cultivars. Coffea robusta. It is grown in the lower elevation between 300 to 610 meters in the districts of southern India. These are those areas where coffee arabica does not thrive. It is also more hardy but yields a poorer quality of coffee. However, it has the capacity to survive in arid conditions and is disease resistant. Coffea liberica. This is a hardy and disease-resistant variety suited to lowland rather than upland conditions. It produces heavy yields of moderate quality coffee. Both Robusta and Liberica are especially suitable for making instant coffee and thus are gaining an importance. Coffea arabica, it is the finest variety of coffee. However, it is also very delicate and vulnerable to leaf and other diseases. It is grown in over 60% of the area under coffee cultivation on slopes ranging from 750 to 1500 meters. Its main varieties are Chicks, Corks, Kents, Margaux, Bourbon, Amarillo, Blue Mountain, etc. It produces superior quality beans, the hectare yield ranging from 500 to 600 kg. GIS of coffee production. As the map clearly indicates, the GIS of coffee production in India. Mostly the three cultivars of coffee are shown. It's Coffea robusta, Coffea arabica, and Coffea liberica. Third one is Chickamauga Arabic coffee, which is grown specifically in the region of Chickamauga district and is situated in the Deccan Plateau, belongs to the Malnad region of Karnataka. Araku Valley coffee can be described as coffee from the hilly tracts of Visakhapatnam district of Andhra Pradesh and Odisha region at an elevation of 900 to 1100 meters above sea level. And the fifth one is Baba Budhangiri Arabic coffee. Is grown specifically in the birthplace of coffee in India, and the region is situated in the central portion of Chikmagalur district. Intellectual property rights. Intellectual property covers a lot of ground and is found in every aspect of the coffee value chain, from patenting coffee makers to trademarking blend names to granting plant breeders' rights. IP's reach is far and influential in coffee history. IP's global history begins with the establishment of patent law in England in 1624 with the Statute of Monopolies. Patents. In Italy, Angelo Moriano was granted a patent for new steam machinery for the economic and instantaneous confection of coffee beverage. The new machine combining water and steam was presented at the Turin General Exposition in 1884 and was the precursor to the espresso machine. German invention of the first dripper and filter also got patent. Trademarks related to coffee. When Minnesota's Dogwood Coffee first began roasting, the company named its cold brew blend as Zamboni. It was a love letter to their staff's enjoyment of hockey and the machines that ruled the rink. We didn't even make it through a full season before we received the cease and desist letter from the Zamboni Corporation, says Dan Anderson, owner of Dogwood. Plants have IP too. Innovation and IP protection in coffee are not limited to equipment manufacturers and roasters, but can even include coffee growers themselves. IP for plants is called plant breeders' rights, which allow breeders to license out a variety to anyone they desire. These are some objectives related to my topic. First one, origin of coffee is tropical America, Brazil, and Ethiopia. The correct answer is Ethiopia. Question number second. In India, coffee was first introduced in which state? Is it Tamil Nadu, B. Kerala, or Karnataka? It was actually Karnataka, where first coffee was introduced. Third, finest variety of coffee is A. Coffea arabica, Coffea snephora, and Coffea liberica. It is Coffea arabica. Cryopreservation of coffee is done at 196 degrees Celsius, minus 196 degrees Celsius, 192 degrees Celsius. It's actually minus 196 degrees Celsius. Caffeine content is highest in Coffea arabica, Coffea snephora, Coffea liberica. It's Coffea snephora. It is Robusta coffee. Efforts to collect and conserve coffee genetic resources were initiated in 1960s to 1970s, or 1860s to 1870s, or 1920 to 1930. It was actually 1960 to 1970.